Okay, first of all, good afternoon. Nice, beautiful, sunny day. And I want to give everybody a warm, congenial Ollie welcome to our 18th iGals series. iGals, by the way, is Irving Goodman Lecture Series. Nice to have an acronym. Anyway, I am Stan Cohen, and I haven't met all of you. I know several of you. And for those I haven't met, welcome to Ollie. Um, I want to also especially thank our committee, of course, that works with fearless leader Jacina, okay, and putting together these Ollie talks. I don't think Linda Yoder is here. She's not here. Barry Edelstein from psychology is here, and I don't see Julie Patrick as well. But they all really spend several hours going over possible candidates. Um, I want to give just a brief history of the series for those of you. There is a program that lists all of our former speakers that you certainly should look at. Um, it's named after Avi Irving Goodman, who in 2007 started this series by deciding that we needed more up-to-date, cutting-edge research in the area of aging. And likewise, he was able to get a $1 million grant from the Osher Foundation, which then transformed us from all, anybody remembers all, Appalachian lifelong learning into OLLI, Osher Lifelong Learning Institute. Okay. After he passed away in 2012, that is when we were able to dedicate the series and name it after him. Oh, and before I forget, which at my age and some of you I think experience this, it's easy to forget. After our speaker time ending around 2-ish, just take a left turn and go straight to the atrium where we will have a reception with some small refreshments. And of course, our guest speaker will be there to follow up on any questions you might have. So let me introduce Dr. Adam Spearer from Johns Hopkins University. And the title of his program lecture tonight is Sleep the Key to Healthy Aging? Exploring the critical relationship between sleep and aging and highlighting the latest research on how sleep disturbances can impact cognitive function, physical health, and the risk of Alzheimer's. And let me spend a moment about I call it his career adventure. Currently, he is professor and vice chair for research and faculty in the Department of Mental Health at the Johns Hopkins Bloomberg School of Public Health. Fortunately, he doesn't have to say all that in an elevator. He has a joint appointment in the Department of Psychiatry and Behavioral Sciences at the Johns Hopkins School of Medicine and is a core faculty of the Johns Hopkins Center on Aging and Health. Okay. And he completed, I'm very pleased to mention, he completed his doctoral studies here at West Virginia University in the Department of Psychology, supervised by Dr. Edelstein. Just a few other facts. He joined Johns Hopkins in 2008, and a year later received a Career Development Award from NIA, and of course that speaks to his cutting edge research achievements to that point and afterwards. He has led or co-led multiple grants from NIA, National Institute of Aging, investigating associations of sleep and or circadian rhythm disturbance with brain health outcomes, including Alzheimer's disease biomarkers. Boy, that is a mouthful. And Of course, describing all of his professional accomplishments would be our hour. So I'm going to pause now and just say we're immensely pleased to have him with us this afternoon. So let's put our hands together to express our welcoming to Ollie.
You got one. Thank you.
Michelle, give this one. You want this back?
Thank you so much, Stan and Ollie. Stan and Ollie. I remember watching Laurel and Hardy as a child. And I want to say it's been so wonderful to be back in Morgantown. I have a special place in my heart for Morgantown and for WVU and the Department of Psychology. And 25 years ago, I believe this month, I took my first courses in that department as a beginning doctoral student with Barry and with Stan. And so it's really very cool to be back here today to talk to all of you. I imagine that Ollie folks tend to be a curious bunch, so you may well have questions. I'll ask that you hold them until the end, if you don't mind, because I get very excited and have a lot to talk about. I probably don't have enough time to talk about everything I'd like to talk about, but I'm going to do my best and edit myself as we go along, depending on how much time we spend on various sections. But I'm going to be talking to you about sleep, and the title is a bit provocative. Is sleep the key to healthy aging? And spoiler alert, I'm not going to give you an answer. Someone told me this talk is, the title is clickbait, but to use a contemporary term, but I will speak to evidence to suggest that perhaps it is. And I'm going to start by giving, if I can figure out where my mouse is, hold on. Oh, here it is. I'm going to start by going to my disclosures because you got to keep me honest. These are some sources of some payments that I like to disclose. Give that a minute. And so this will not be news to you, but the US population is aging. All right. And you can see that in 2012, there were over 40 million people aged 65 and older in the United States. If you fast forward to 2050, can you see my cursor? I don't think you can. Is this Oh, that's okay. Okay, great. And if you fast forward to 2050 on the right side of that figure, you'll see that you get above 80 million adults age 65 and older. Thank you so much, Michelle. And then the figure on the right side is basically telling the same story, that the demographics are shifting such that there are far more older adults among us than previously. But you might ask, well, what does this have to do with sleep? And some of you say, well, I know exactly what that has to do with sleep because I'm of a certain age and I can't sleep. But others might wonder. So sleep disturbances are quite common among older people. And poor sleep is more and more being linked to poorer health outcomes among older adults, including cognitive decline and functional decline and Alzheimer's disease. And late life sleep disturbance, in fact, sleep disturbance across the life course is modifiable. And because it's modifiable and it's linked to these poor health outcomes, we wonder if maybe it's a modifiable risk factor that we might leverage to promote better health in people as they age. So how common are sleep-related symptoms among older people? So this is a study that was done in over 9,000 community-dwelling older adults back in 1995. And it, I think, illustrates this nicely, that these complaints are sleep problems are quite common. So in this sample that had a mean age of 74, more than 80% reported that they have one of the various sleep-related complaints, sometimes or most of the time. And then they looked at the proportion of the sample that reported having a complaint most of the time. And you can see here, it's about 20% had trouble falling asleep most of the time. they called this a chronic sleep complaint. 30% had trouble with waking up during the night most of the time. That is the most common sleep-related symptom that older adults report. Waking up too early, almost 20%. A quarter were sufficiently sleepy during the day that they needed to take a nap. And when they looked at these and other sleep-related complaints, almost 60% had one or more. So sleep disturbances are common in the general population of older adults. But why? Well, we think that in part it's due to what we are calling normal age-related changes in sleep. And I'm saying what we think, well, I'll explain why. So if you look at people across the life course, starting over here at age 5, moving all the way across here to 85, you'll see that there are some changes that, or at least this summary figure comes from a series of cross-sectional studies, snapshots. They didn't follow these people over time. So there's a bit of a leap about the extent to which these are changes or related to, well, I don't have to get into the weeds here, but basically sleep gets shorter, okay? Sleep duration decreases. You have more what they call WASO, wake after sleep onset. So that's, you fall asleep and then you find yourself awake, more often than not before you'd like to be awake. There are decreases in REM sleep, rapid eye movement sleep, that occur across the life course. And that is the stage of sleep in which we do a lot of our dreaming, and it has implications for the storage of memories, as does slow wave sleep. So slow wave sleep is the deepest stage of non-REM sleep. So your eyes aren't doing the rapid eye movement thing, but your brain is exhibiting these slow rolling waves of electrical activity, okay? And it's interesting, that also plays an important role in memory consolidation. But if you note, that is perhaps the area in which we see the biggest change with age, significant reductions in slow wave sleep. And as I'll mention in a little bit, we think that has implications for Alzheimer's disease. And then, so we have less REM, we have less slow wave sleep, our sleep is shorter, we have more sleep fragmentation, we're waking up more, and we have more time spent in lighter sleep stages, stage 1 and 2. So, and the reason I said in normal aging is these findings remain when you look at people who have remained relatively healthy throughout life. They're not accumulating chronic conditions at a significant rate. They're relatively healthy people. So this alone does not, we think, account for the very high prevalence of sleep complaints among older people. Rather, we think that medical conditions, and to a degree psychiatric conditions that can emerge as we go through life, combined with the medications that people take for these conditions, we think that those things play a significant role in ramping up sleep-related complaints. Also, as we get older and we're not able to move in quite the same ways as we did when we were younger, there can be that decrease in physical function can make it harder to get up and about. More sedentary time can lead to, first of all, we know exercise is good for sleep, okay? As long as it's not too close to bed, we think. But also, if there's more opportunity for sedentary time, people might be more likely to fall asleep, right? And that can interfere with nighttime sleep, because when you sleep during the day, you reduce what we call the homeostatic pressure to sleep that builds up from the time that you wake up in the morning until the time that you go to sleep, and then it starts to decrease again, right? Psychosocial changes, including caregiving for a loved one. We know that family caregivers, particularly family caregivers of people with chronic medical conditions such as dementia, can really experience sleep problems as a result of either their stress related to the caregiving or the sleep problems of the person with dementia for whom they're caring, for example. There's also bereavement that happens as we get older and we experience more losses. That can affect sleep. And then another one that can have potentially beneficial effects, but also not so beneficial effects, is retirement. it turns out people do, some people do get more sleep when they have more time to sleep, but that greater opportunity for daytime napping, for example, in some people it doesn't interfere with their nighttime sleep, but it can if you do too much of it through the same mechanism that I described with the homeostatic drive reduced. We have all of these processes that go on in our bodies on a roughly 24-hour basis, okay? This refers to changes in core body temperature, expression of genes, release of hormones and production of hormones. All of these things will sort of cycle on a 24-hour basis, and we call them circadian rhythms, particularly when they are things where the rhythms will persist even in the absence of stimuli that basically synchronize them with the external environment, like bright light. Anyway, as we get older, one of the most common observations is that we get sleepy earlier and go to bed earlier, and we wake up earlier. That is not a hard and fast rule, but it is a very common phenomenon among older people. And some older adults will say, well, I have a sleep problem, even if they're getting 8 hours of good solid sleep, because they find that they are sleeping at the wrong time. So they don't like the fact that, even if they're getting 8 hours of sleep, they're waking up at 4 a.m. That could be a bit of that. We call that a circadian phase advance. Anyway, so those circadian rhythm changes can affect sleep and lead to sleep-related complaints. And sleep disorders become more common as we get older. And I'll talk a little bit about sleep-disordered breathing as we move along. That's a common one. Sleep apnea. You've probably all heard of sleep apnea. But I want to tell you a little bit about some research that I did that came from the grant that Stan graciously mentioned. And thanks for that very kind introduction. I really appreciate it. To look at sleep in relation to functional decline and disability in older people. And we did a study that we published, I can't believe it was 2012, but I found this was a meaningful study for me. My primary mentor on this study was Christine Yaffe at UCSF, where I did a postdoc. But there was evidence before I did this study linking poor sleep to functional limitations and disability in older people. However, most of the studies were cross-sectional, where they had the sleep and the functioning measured at one time point in a snapshot. One of the problems with that is that, of course, it's hard to tell. There's no temporal separation between the predictor and the outcome, so you can't tell if it's the chicken or the egg. And something that gets us a little closer to that, although it doesn't get us all the way there, is a longitudinal study where you can say, at time A, sleep disturbances predicted change in function. That gets us a little closer, because one of the things that's required for causality is that the cause come before the effect. Doesn't fully answer the question, but it gets us there. Also, a lot of those studies that existed had looked at self-report measures of sleep, where you ask people about their sleep. People don't necessarily give 100% accurate information about their sleep. I couldn't tell you exactly when I fell asleep last night or exactly when I woke up. And when you ask people to do that, you can get some error, particularly when you're studying older people who might be more vulnerable to Stan reference to memory concerns. So that is something to keep in mind. So we needed to have studies that used objective measures of sleep. Also, the studies had used self-report measures of functioning, which can be very helpful, but are not the same thing as getting objective measures of functioning. Asking someone about whether or not they can do something or how strong they feel versus actually measuring it. So in this study, we decided to look in a cohort of older women, community-dwelling older women, what the association was of objectively measured sleep with new difficulties with instrumental activities of daily living. And I'll tell you which ones we measured momentarily. And with decline in performance-based measures of physical function. And we studied 817 women in a study called the Study of Osteoporotic Fractures, which recruited mostly white women, 9,700 of them approximately in 1986, white women are more vulnerable to osteoporotic fractures than non-white women. So they focused on white women, but then they added some Black women later in the study. But the inclusion criteria was age of 65 and older, the ability to walk, to ambulate without assistance, and to not have a history of bilateral hip replacement. So pretty broad inclusion criteria. And so they wore these wearable devices. There was a picture of one of them on this first, see it here. It's like a wristwatch-like device that you wear on your wrist. And many of you probably have a Fitbit or an Apple Watch. Anyone wearing one? I see lots of hands. So with an accelerometer in them. So way before those became the thing, sleep researchers dating back to about the 90s had been, 80s or 90s, started using them, leveraging the fact that we know that we move less when we're sleeping to maybe get a sense of whether or not someone is asleep. And these devices use algorithms that have been validated against the gold standard sleep measure, which is polysomnography, a full sleep study, to differentiate sleep from wake. So in year 16 of this study, the women wore this device for about a week. And then at year 16 and 20, they completed these functional measures, including self-report of IADL difficulty. In this case, difficulty with grocery shopping, heavy housework, or preparing meals. And then they completed performance-based measures of grip strength and gait speed, okay? And so this is their characteristics. There were about 82, about 13% non-white, 40% had a greater than a high school education, et cetera. So, and what we found was, I'll give you the punchline here. They had about 6.8 hours of sleep per night on average. And they were awake after they fell asleep. They were awake for a little over an hour, 66 minutes on average. And they slept. Sleep efficiency is the percentage of time in bed that you spend asleep. And they had an average sleep efficiency of 80%, which isn't too bad. It's not great, but it isn't too bad. Anyway, so all of these women were free of IADL problems at baseline. All right, they didn't have any. But 5 years later, an average of 5 years later, 41% of them reported that they had a difficulty with one of those IADLs. And I'll give you, there was no association between the sleep parameters and gait speed, but I'll show you some interesting results here. So compared to the women with the longest sleep, the 25% of the women with the longest sleep, those who had the shortest sleep had double the odds 5 years later of having a new problem with their instrumental act, with at least one IADL, okay? When we looked at wake after sleep onset, you fall asleep, but you wake up afterwards, we didn't find any associations between the amount of wakefulness after you fall asleep and new IADL problems. But we found that compared to the women, the 25% of the women with the highest sleep efficiency, who spent the most time in bed asleep, those who were in the top 50% of, the bottom 50%, I should say, of sleep efficiency, each had, 70% greater odds, roughly, of having a new IADL problem. And when we looked at significant decline in grip strength as being in the 25% of those, the highest 25% of weakening over 5 years in your grip strength measures. So, and compared to those who had the longest sleep duration, those who had the shortest had double the odds of significant decline in grip strength. And we also found that wakefulness after sleep onset was strongly associated with a greater odds of declining grip strength, as was sleep efficiency. If you had the lowest sleep efficiency, you had about double the odds of significant weakening. 4.75 kilograms of weakening is how we defined it over 5 years of your strongest grip that you can do. You know, when you go to the doctor, they have you squeeze the hand dynamometers to test your grip strength. That's the measure. So I'm going to quickly tell you, we also wanted to look, well, what if we look in the same cohort of older women at how sleep measured objectively is related to nursing home placement or placement in a long-term care facility of some sort? And so in this study, we had 1,664 women to study in this analysis. All were community dwelling at baseline. 5 years later, 4.3% were in a nursing home, and 7.6% were in an assisted living facility type of arrangement. And we found that greater WASO, wakefulness after sleep onset, was associated with almost 3 times the odds of being in a nursing home, which is really pretty remarkable 5 years later. And that was after accounting, I should mention in the other study, we did a similar adjustment statistically for age, race, BMI, MMSE, that's a cognitive performance, number of IADL impairments or mobility impairments at baseline, and other variables. We similarly found that having low sleep efficiency was associated with a much greater odds, over 3 times the odds of being in a nursing home. And we found some similar associations with being in an assisted living facility 5 years after the baseline measurement. And you see the same thing for sleep efficiency. So this was I was pretty excited about these papers when they came out. They were the first studies to show links of objective sleep measures with these sorts of disability-related outcomes. And one of the things that comes up, though, is, well, how is sleep doing this? What is the mechanism by which sleep might lead to these outcomes? And it could be, even though we adjusted statistically for medical comorbidities that people had, there could be some that we didn't capture, or some sort of subclinical, we might call it, physiological vulnerability that we didn't capture related to perhaps the coming onset of those disabilities. And then also, we think inflammation, you hear a lot in the news about inflammation and how it's bad news when it's chronic and low grade. It helps us in the short term, but we know that insufficient sleep can lead to, can increase inflammation. And inflammation is also associated with a range of poor health outcomes that might lead to functional decline, including weakening of the muscles, sarcopenia, as it's known, and all sorts of other chronic health conditions. So maybe that's a mechanism. But what this was exciting to me because it raised the possibility of a role of maybe leveraging sleep. Perhaps we can improve sleep to prevent disability and nursing home placement in some older adults. And I'll talk a little bit about how we might do that later, but the Q&A would be a good time to talk more about that. Now I'm going to shift gears. While I was doing this work for my Career Development Award, I became interested. There was a burgeoning area of the literature on sleep and Alzheimer's disease. And Alzheimer's disease, or AD, and dementia, a few words about that. So Alzheimer's disease is the most common cause of dementia, and it's defined by these extracellular, so outside the neurons in our brains, beta amyloid plaques, or A beta plaques, that appear early in the Alzheimer's disease course. And then within the neurons, there are these intraneuronal neurofibrillary tangles that develop as the disease has progressed. And as I said, it's the most common cause of dementia. In 2024, the Alzheimer's Association has estimated that there are 6.9 million adults in the US age 65 and older with Alzheimer's disease. And the prevalence of Alzheimer's disease will quadruple from the year 2000 to 2050 globally. That is a staggering statistic. And we are not the only country in which we're seeing these sorts of changes in demographics, okay, in aging. And there's no cure for Alzheimer's disease. And there are some treatments that are on the horizon or have just emerged that show some promise, but there's nothing that has really led to a truly substantial disease-altering consequence. So for that reason, prevention is key. And people have started to focus on sleep as one of those factors that might be related to dementia or Alzheimer's disease prevention. So when people used to talk about neurodegenerative disease, including Alzheimer's disease and sleep, they used to think about it in this way, that their neurodegenerative disease changed the brain, which would lead to sleep-wake disturbance. And this is consistent with what I referred to earlier about dementia family caregivers and the observations that they've had and reported to their family doctors about the significant sleep disturbances that the people they care for have been exhibiting, for example. And we hear about this a lot clinically and just anecdotally. If any of you have friends who have been family caregivers, you might have heard about this. And this is because, these are really, when you compare the sleep of older adults with Alzheimer's disease to the sleep of older adults without Alzheimer's disease, you see something interesting. You remember that I described how across the life course, our sleep gets shorter and more fragmented, and we have reductions in REM sleep and slow wave sleep. Well, guess what? When you compare older adults with Alzheimer's disease and other forms of dementia to those who are to older adults without, you see the same sort of differences. The sleep of those with dementia is shorter and more fragmented. You see lower sleep efficiency, spending a smaller proportion of time in bed. So, and then reductions in slow wave sleep and REM sleep. And greater dementia severity is associated with greater sleep fragmentation. And it's fascinating that there are these Alzheimer's mouse models that are genetically programmed essentially to develop A beta plaque. And what's been shown is in an AD mouse model that develops amyloid beta plaques, as you just observe them over time, and as the plaques develop, you see alterations in the sleep-wake cycle. They start to change. And what was very interesting here is these authors did an immunization of these mice with the A beta 42 peptide, and that active immunization prevented the deterioration of these sleep-wake cycle, suggesting that yes, that it was the A beta that was causing the problem. Now, so this is the way people had typically been thinking about it, as I said. In 2009, however, there was a fascinating study that sort of made us all stand up and pay attention if we were studying sleep and Alzheimer's disease. Kang et al. At WashU in St. Louis, Washington University, showed that if you sleep deprive an AD mouse, one of these mice that's programmed to develop Alzheimer's plaques, you get substantially greater amyloid plaque burden after the sleep deprivation than if you don't sleep deprive them. And that was the first evidence of a causal link between sleep loss or sleep deprivation and Alzheimer's disease. So that was really sort of mind-blowing. And I was I'd been at Johns Hopkins a year at that point, and I partnered up with some colleagues, and we wanted to really dig into this. And one of them, this is a study by Masashi Tabuchi, led by my colleague Mark Wu is the senior author. He showed in a Drosophila model of Alzheimer's disease. Drosophila is, a fruit fly model. And did you know that, first of all, a lot of how we sleep is actually inherited from Drosophila. The term that is used is conserved, right, from Drosophila. And we've learned a lot about the biology of sleep and the genetics and epigenetics related to sleep and the clocks in our brains from Drosophila. Anyway, I told you I could talk a lot about this stuff. And fruit flies, like us, sleep at night. Mice sleep during the day. Okay, so at night, if they shook the tubes in which the fruit flies were living, they live in tubes and there's food at the bottom, and they basically walk up and down when they're awake, and they eat. I mean, it's really very similar to a lot of us. When they shake the tubes they live in while during that nighttime period when they were sleeping or supposed to be sleeping, you see this green. These are immunostained slides of the fruit fly brains. You get much more of the green, the light green, which is the amyloid, if you shake those tubes and keep them awake. If you but then people say, well, could it be just the shaking the tubes that's the problem? Well, it is stressful to have your house shaken But if you shake the tubes during the day when they're awake, it doesn't do anything. So really, they see, you don't get that sort of amyloid deposition. So we really think it is the sleep loss that is causing the problem. So this really, this was the conventional view that neurodegenerative disease and brain changes lead to poor sleep. But then this led us to wonder, the field, us, capital U, the field, could it in fact be that sleep-wake disturbance is leading to Alzheimer's disease? And so my colleagues and I started to do work in this area. This is a study that we published in 2013 in JAMA Neurology. This was a big paper for me at that point. I was an assistant professor, and we studied 70 older adults in the Baltimore Longitudinal Study of Aging. This was in collaboration with researchers at the National Institute on Aging Intramural Research Program. They shared data with us, and we worked together on this. They had a mean age of 76. They were 47% were women, almost 20% African American. This is a very highly educated group in this study. They're like Ollie people, right? 17 years of education on average. That's a very high level of educational attainment. And they had high mini-mental state exam scores. The highest you can get is a 30, and the average was 28.9. And they self-reported their sleep. And then within 5 years of that, they had PET scans of their brains that actually show in vivo, in living people, how amyloid is binding in the brain. And what we found was, so the DVR is the distribution volume ratio, which is just a continuous measure of amyloid burden. And we looked at two different measures of amyloid burden. One that looked across many different parts of the brain, and we called that the cortical DVR, across the cortex. That's sort of a global summary measure of burden. And then we also looked at the precuneus, which is a region in which A beta aggregates early in Alzheimer's disease. And after adjusting for age, sex, race, depressive symptoms, BMI, a genetic factor, ApoE, the ApoE4 allele is associated with risk for Alzheimer's disease, and cardiovascular or pulmonary disease and use of sleep medications, reports of shorter sleep duration were associated with greater amyloid deposition in this global summary measure of beta amyloid burden, and in the precuneus, which is that region that's vulnerable early in the AD course. We also saw that reports of poorer sleep quality, worse sleep quality, were associated with greater amyloid deposition in the precuneus. And if you look going from left to right, these vertical, first of all, these vertical columns are, they're these slices, horizontal slices of brain that stack on top of each other. But if you go from left to right of these colorful columns, that's going from the longest sleep duration reported to the shortest. And when you see more of the orange hot colors, that means greater amyloid deposition. So this is pretty exciting to us. It was an early study, and it was very important to my career development. But we continue to do other work in this area, looking at this A beta protein. And I asked them, well, what variables do you have that way predate the amyloid scans that were collected a long time before people had any amyloid scans done? Because the more we can separate things in time, the more we can potentially speak to a causal association, or at least the potential for one, right? You can only prove that something actually causes something if you experimentally manipulate it. And we can't sleep deprive people for years and look at what happens, right? There are some ethical problems with that. So we have some, methodologic limitations that are good to have imposed on us, right? So observational data where we measure people's sleep and follow them is about the best that we can do if we want to look at this in naturalistic sort of settings. So in these folks, also in the Baltimore Longitudinal Study of Aging, they were asked a single question, very early in, by the way, the Baltimore Longitudinal Study of Aging started in 1958. It is the longest running study of aging in the United States, if not the world. Anyway, a little bit of information there. But they were asked, do you often become drowsy or fall asleep during the daytime when you wish to be awake, e.g., falling asleep, watching TV, or reading? Now, when we compared and we call that EDS, excessive daytime sleepiness. So they were asked that question, and then they had amyloid PET measured 15.7 years later on average, okay? And the range was, it was from 7 years after they were asked that question to 25 years. And what we found was, compared to those who said, no, I don't have that problem of often becoming drowsy or falling asleep when I wish to be awake during the daytime, after accounting for age, sex, education, and body mass index, compared to those who said, no, I don't have that problem, those who said, yes, I do, had 2.75 times the odds of being positive for amyloid in their brains. This was a very simple study with one question, right, on the predictor end, and just a dichotomous outcome, positive for amyloid in the brain or negative for amyloid in the brain. And so this was a pretty surprising result. And we don't know, to interpret this is a little bit difficult because we don't know what the cause of the excessive daytime sleepiness is, right? People can be sleepy during the day and doze off for a bunch of reasons, but probably something going on with their sleep at night, right? And one of the things that you will often hear about, and if you've gone to the doctor and complained about being sleepy during the day, they might have said, maybe we should get you a sleep study to see if you have obstructive sleep apnea. That's a very common condition, right? So, and that falls under the broader category of sleep disordered breathing. So sleep disordered breathing is characterized by these repeated interruptions in breathing during sleep. If you stop breathing, it's an apnea. And if you have a significant decrease in your breathing combined with a blood oxygen desaturation of a certain amount or an arousal from sleep as a result, that's a hypopnea, okay? And obstructive sleep apnea is the most prevalent subtype or cause of sleep disordered breathing. And the risk factors are older age, higher body mass index, greater neck circumference, and being male. Those are some of the biggest risk factors. And it's measured by polysomnography. And you get wired from a lot of electrodes on your head and wear something on your face to measure your breathing. They actually do these studies a lot of the time without electrodes. If you've been sent home with one of these, it might have just been a nasal cannula and a pulse oximeter and maybe some bands around your chest to have it measured. Anyway, there's an apnea-hypopnea index that is used to gauge apnea, sleep disordered breathing, I should say, severity. And it's really prevalent in older people. If you look from age, and it depends where you look for the source of this information, but anywhere from 20% of adults age 60 and up to 62% of adults 65 and up, okay? And when I was a postdoc working with Christine Yaffe at UCSF, I published this paper looking in that same cohort of the study of osteoporotic fractures. This was in the Journal of the American Geriatric Society. And it was about 448 women in the SOFT study, age 83, who completed in-home polysomnography. And their average AHI, that's that apnea-hypopnea index, was 15.5. Now, an AHI of 15 or greater is moderate to severe sleep disordered breathing. So it's at least moderate. And these women were doing quite well on their MMSE on average at 28. And 11% of them had that ApoE4 allele that I mentioned is linked to Alzheimer's disease risk. And what we found was, if you look at this table here, I'll just distill it. On the mini mental state exam, which is a measure of cognitive performance, it's a broad, fairly blunt measure, but it's used widely. So it was available in these data. Each standard deviation increase in the AHI, in apnea or sleep disorder breathing severity, was associated with a 40% greater odds of having impairment on that test, which was measured by being more than 1.5 standard deviations below the mean on that test. And we found that people with severe severe apnea, for example, that's an AHI of 30 or greater, had 3.4 times the odds of having significant impairment on that test. I could show you more results, but I think you get the point. We didn't find anything for this other test, the trail making test, although I have some ideas as to why that might be the case. We can talk later if you like. What was interesting, when we looked at the women for whom there was genotyping, where we had genetic data, and we compared the association between the AHI and impaired performance on the mini mental state exam, the association was much stronger among those who were E4 carriers, interestingly. So there's something about that genetic, and by the way, E4, ApoE4 has been linked to a greater risk of sleep disordered breathing, in addition to a greater risk of Alzheimer's disease, sorry. So it's an interesting and potentially complex situation. But then I was on this paper, I'm right in the middle, which is not the best position on the paper. You want to be like first or last, right? But I'm in the middle, and I'm okay with that because it was published in JAMA. JAMA. So that was exciting. But this was led by Christine Yaffe, who has been a terrific research mentor to me. But this was 298 women in the same study who had a median AHI of 10, and 35% had sleep disordered breathing if you use this moderate sleep disordered breathing cutoff to identify being positive for SDB. And what we found was, if you compared those who did not have moderate to severe sleep disordered breathing to those that did, those who did had 1.9 times the odds of having mild cognitive impairment or dementia 5 years later, okay? And it seemed like really desaturations in oxygen levels were driving this. I mean, it's not that surprising that significant desaturations in your blood oxygen are not potentially good for your brain, right? So we think that's the mechanism that might have accounted for this. So that was a little departure into sleep disordered breathing. I want to get back to talk about sleep duration. We did a study, again, in the Baltimore Longitudinal Study of Aging, where we were curious about sleep duration. And I can talk towards the end if you have questions about sleep duration during the Q&A. But there were 122 participants in the Baltimore Longitudinal Study of Aging, and they were age 67 on average when they reported their sleep duration. And then they were followed over many years with repeated MRI scans of their brains. And I thought, well, we should take advantage of that and see how sleep duration is related to changes in brain structure. And so we wanted to look at gray matter atrophy. So these are repeated measures of cortical thickness. And we only used data from visits at which participants were cognitively normal, okay? So that's important to keep in mind. They tested normally cognitively throughout all of this data that went into the study. And what we found was, if you use 7 hours of sleep as the reference category, and by the way, for older adults, 7 to 8 hours is the recommended sleep duration, all right? 7 to 8 hours. If for adults who are a little younger, before you get to age 60 or 65, I forget which age they used as the cutoff. I can look that up later if you like. It's 7 to 9 hours for younger adults. It's 7 to 8 for older adults, and we can talk about why in a minute. But those with the shorter sleep duration had a higher had greater cortical thinning over the subsequent period of evaluation after they were followed in these temporal and frontal regions of the brain, and almost entirely on the left side, and we don't know why. But it was left hemispheres where we saw this. And then, more than 7, if people reported more than 7 hours of sleep, they had more rapid cortical thinning in these frontal regions, which was interesting. So more is not always better, although it is, and so that's why they don't recommend, not from this study, plenty of other studies to suggest that staying in that 7 to 8 hour range and not doing more than that is better for your brain health if you're an older person. However, we don't know, we can't figure out exactly what the causal mechanism would be linking more sleep to bad brain health outcomes. We can talk about that more. So this is a pretty picture showing the same thing that I just talked about. And so it looks like probably this is the right way to think about it. There's a bidirectional link between sleep and neurodegenerative disease. But how does this happen? Well, I like this figure from Yoel Ju and colleagues. So what they believe, and what I believe, is that poorer sleep quality leads to reductions in slow wave sleep. And we get less of that as we get older anyway, right? And we think that slow wave sleep, one of its consequences is that it actually helps decrease the connections between our neurons in our brains that form over the course of the day. You get up in the morning, you start walking around, you read the paper, you learn something, some synapses connect, and later on that gets transferred to longer term memory after you have some good sleep. You know, you engage in conversations, you navigate your world, lots of synaptic connections are made. And some of those connections become stronger and stronger as the day goes on. And we think that actually too much synaptic strength, too much of a propensity to fire is not a good thing. That actually too much of that electrical activity is bad, and neuronal activity is neurotoxic, and that slow wave sleep actually downscales those connections, decreases the synaptic strength. Not only does that serve the function of basically wiping the slate clean to allow for new learning the next day, but we think that it also reduces production of amyloid beta, which we all produce in our brains. But we think that without the slow wave sleep to downscale it, you would get excess production of A beta. And then this leads to, and so you can see that here, neuronal activity leading to A beta release, leading to more A beta in the brain and more risk of plaque formation, and then symptomatic Alzheimer's disease. And we think that, as I've described previously, greater amyloid deposition in the brain deteriorates sleep quality. We think that the amyloid plaque itself messes with sleep. And then we've got sleep disordered breathing, which can lead to amyloid deposition in Alzheimer's disease by disturbing sleep quality and eroding slow wave sleep, but also through inflammation. Remember inflammation we talked about earlier, and hypoxemia, which we think that's another path by which it can lead to amyloid production and aggregation. The other thing that's interesting is in 2013, there was a paper that came out showing that during slow wave sleep in mice, there appears to be a dishwasher-like effect in the brain, whereby the space between our brain cells actually increases during slow wave sleep, which facilitates the flushing out of toxins. It sounds like science fiction, but it actually, there is evidence for this phenomenon. And it's mediated by these aquaporin-4 water channels in our brain, okay? And there's a lot of research going on in this area. But that's another mechanism. Maybe through sleep, specifically slow wave sleep, we are cleaning out toxins. Now, the other thing I want to show, and I'm sort of closing with this, and I didn't even leave as much time as I hoped, but there's a reception. We can talk during that. This is a study my colleague and collaborator, Brendan Lucy, published recently that showed actually that so this says 38 cognitively unimpaired participants aged 45 to 65, they were randomized to receive either placebo or this drug, suvorexant, which is a dual orexin receptor antagonist. It blocks orexin, okay? And what they showed was that in those who received suvorexant, and this drug is a sleeping aid, it's a sleep aid. It's one of the newest forms of sleep aids, all right? These dual orexin receptor antagonists that we think could have a safer side effect profile in older people than some other medications. A lot of sedative hypnotic medications that are taken for sleep are actually not recommended for any kind of long-term use, particularly in older people because of risk of falls and fractures and confusion and delirium and things like that. But this drug in these relatively healthy young people actually decreased phosphorylation of tau, which is to say there was less of what we think goes into, of the ingredient that goes into neurofibrillary tangles, all right, as a result, and amyloid beta concentrations were lower in the central nervous system. What they did in this study is they continuously monitored, I think it was over 36 hours. Yes, they had a catheter put in their spinal column, and they measured their CSF, cerebrospinal fluid proteins of tau and amyloid continuously. And they showed that there was this positive effect of this drug. So that's something that could be, keep your eye on that in the news media and so on. This study was in the news a lot. I do want to mention, in general, sleeping aids, medications are associated with a lot of poor outcomes in older people. And if you like, I can tell you a bit about alternative approaches to improving sleep. But, so I will ask you, is sleep the key to healthy aging? I think that it seems to play an important role. Then again, I'm biased. I talk about sleep all the time. It's what I study mostly. And I enjoy it. I mean, who doesn't enjoy some good sleep? So, I'm a bit biased, but I think it really is important. And, there's more and more literature coming out all the time to suggest that is the case, particularly for brain health and other aspects of health as we age. So I want to say thank you to all of you, to the NIA who funds my work, to my co-authors, collaborators, research staff, trainees, participants in the study of osteoporotic fractures and the BLSA, Baltimore Longitudinal Study of Aging. And thanks very much to Stan and Barry and the Ollie folks. I wouldn't be here talking to you today if Barry Edelstein hadn't been I hadn't started working in his lab 25 years ago, roughly. And so it's really a pleasure to be back here giving this talk to all of you. Thank you for having me. And I'm happy to take any questions. I see that someone online has a chat, intervention at fragmentation sleep stage. Do you want to say more about that? I think that maybe it's getting at, yeah, go ahead.
Yeah, I just want to find out anything can be done to defragment the sleep stage. Okay, I'm a medical technologist, public health person, and I do get fragmented sleep. Okay, I do wake up. So is that compensated by an afternoon nap, or what? I mean, how much is the quality of the sleep? How many hours do you need and all that? Okay, so.
Thank you. These are great questions. And so we think that once you get to age 60 or 65, that 8 hours is really 7 to 8 hours is what you want to go for in terms of duration. We don't yet, by the way, napping is a fascinating area to study. And if you look, I was involved in what's called a meta-analysis related to napping and cognition not too long ago. You see mixed results because I think the reason is people nap for different reasons, right? People can nap because they are really fatigued and they feel they have to lie down and they fall asleep. They can nap because it's part of their culture. In many parts of the world, there's a siesta, right? After lunch. Not all naps are created equal. There are some very long naps, and there are those cat naps, those 20-minute naps that we think are probably the best if you're going to nap, because it's unlikely that you'll get into deep enough sleep that you'll have a lot of sleep inertia and grogginess once you wake up. We don't know yet how much offsetting nocturnal sleep fragmentation with daytime napping to compensate might be beneficial for brain health. It's a great question. In terms of interventions to decrease sleep fragmentation, If there is a disorder like sleep disordered breathing, that is something that is going to, by definition, fragment sleep. The two consequences are sleep fragmentation and hypoxemia physiologically. So, and sometimes we might not think that we have sleep disordered breathing, but it's hard to know. We think we might just wake up because we have to use the bathroom. That's another thing that becomes very common, right? Waking up several times to use the bathroom at night. But it also, so just as an example with sleep disordered breathing, it's not uncommon for people to say, you know, now that I treat my sleep disordered breathing, I find that I'm not waking up to use the bathroom as much. And part of what we think happens is sleep gets fragmented by the obstructive sleep apnea. People have a microarousal, they wake up a little, and they feel the pressure on their bladder, and they get up and go. Whereas if they slept through it, it wouldn't be an issue. So it's a tough, one of the other things that we can do to try to promote consolidated sleep is to avoid naps. As I mentioned, anything you do to decrease the homeostatic drive to sleep will potentially interfere with your, getting good consolidated sleep at night. Those are a couple of answers that are not necessarily satisfactory, but I hope they're a little helpful. Roy, anyone, before I answer another one here, anyone have a question in the room? All right, first this gentleman, yeah. Oh, wait, there's a microphone coming, hold on. There you go.
Have you seen that research has shown any long-term effects of COVID vaccination, the RNA vaccination, mRNA, on sleep patterns?
I have seen research not on vaccination, but we're seeing all sorts of neurologic consequences of COVID and long COVID. And I believe people have talked about sleep. I haven't studied that very much. I haven't seen, but I'm not aware of COVID vaccination and those outcomes.
Okay, follow up, a different subject. If you have a sleep cycle, you can have a sleep cycle of about 3 hours, can't you?
Well, it's about 90 minutes on average.
Okay, so you go through more than one cycle at night. What's the difference between the cycles in terms of quality and value?
Oh, it's a good question. Yeah, so you have several cycles of sleep as the night progresses. So, and they actually, the proportion of time spent in different sleep stages changes as a function of the time in the night. So talking again about homeostatic sleep drive, one of the ways that we think that we're able to gauge how strong your sleep drive is the amount of slow wave sleep you get early in the night. Early in the night is when you get most of your slow wave sleep, okay? And then as you get more and more of that, you spend less time in slow wave sleep, and you spend an increasing amount of time in REM sleep. And in the morning, by the time you wake up, you've recently had the most of your REM sleep. So the composition of the cycles shifts over the course of the night. The reason that we think slow wave sleep must be really important because it's the first thing that it'll be prioritized by your brain earlier in the night.
Last night I took a sleep aid for the first time. This have to be closer. Okay. And so, I mean, we're here today and I'm nodding off during your, you know.
Thank you very much.
Hopefully I didn't snore. I don't know, but
I appreciate you mentioning the sleep aid.
I can blame it on my first trial of the sleep aid. But like my issue is generally speaking, getting to sleep. Like I can go to bed at 11 or 12 at night and I'll lie awake. I'll still be awake at 4 in the, just in bed, rolling around. And I've tried brown noise, and all kinds of things. And it doesn't happen every night, but it happens often enough that it's an issue for me. And does that mean, you're saying that the slow wave sleep that happens or earlier at night when we go to bed.
It typically does, yeah.
So
you get more of it earlier.
Well, so would I likely be not getting much of that, or would I just be getting that later and then not getting as much of the others?
That's a good question. And there is a literature on how these medications. So one of the things that's very important to note is not all, two people, right? One who sleeps without the help of a sleep aid, one who sleeps with a sleep aid, their eyes might be closed. They might otherwise look very similar, but what's going on under the hood, if you like, is different, right? And I actually can't speak to the specific changes there are differences by different medications, okay? And I'm not a pharmacologist,
Mhm.
but different agents in different classes actually have different effects on sleep architecture, the time you spent in different sleep stages. For example, there is a medication that's often used for sleep, even though it's not, it's actually an antidepressant called trazodone that's widely used for sleep because it has relatively benign, side effect profile. It's not a very good antidepressant, but it helps people get drowsy and fall asleep. It's also not habit forming, and some of these sleep medications can be, right? So it's used not uncommonly. And one of the things that we know it does, it increases slow wave sleep, which is an interesting phenomenon. So we're actually doing a randomized trial right now at Johns Hopkins. The study is called REST, and it's an acronym for something, but it's a cute acronym. But it's a randomized trial of trazodone for people with mild cognitive impairment to look and see, it's a placebo-controlled study, the extent to which trazodone might boost next day functioning and potentially affect Alzheimer's disease biomarkers in that population.
Would this trazodone be one of the suvorexants you were talking about?
No, it's not. Different
Okay.
class. So that's suvorexant is a DORA, dual orexin receptor antagonist. Now, I want to give a big plug for cognitive behavioral therapy for insomnia, or CBTI. It is not every day that the American College of Physicians says this is the first line treatment for a condition, and it's a behavior therapy developed by psychologists primarily. And so it's a big deal. They have said this is the first line treatment before medication for anything for chronic insomnia. And there's a question about it. Is it about CBTI or? Yeah, go ahead.
Okay. There was a time when I had a monkey brain. I mean, I just couldn't, I worried because of what was happening with my children. Okay. And so I learned meditation,
Oh, yes.
cognitive. So what I did was I started breathing in. Let's see, what was the first word I used? Peace. I breathed in and thought the word peace. And then I breathed out and thought the word peace. So if I was saying those words, I couldn't be worrying about everything else.
Yeah, so if I can just say something about that. So
Sure.
that is a different sort of an intervention. I want to talk about that briefly if there's time and I don't get the hook. But cognitive behavioral therapy for insomnia is different. It really teaches it basically dictates that you change things in your behavior that really can make a difference in your sleep. And there's not really time for me to go into the details here, but there are practitioners all over the United States. And if you go to the Society for Behavioral Sleep Medicine online, Society for Behavioral Sleep Medicine, this is a non-drug approach that is wildly effective across the life course, including in older people. There was a randomized trial, to get to your excellent question about, or statement, of mindfulness meditation in older adults who complained of poor sleep quality as measured by a score on the Pittsburgh Sleep Quality Index. Go Pittsburgh, it's right over there. So, and that randomized trial showed improvements when they taught mindfulness meditation at senior centers, and I actually wrote the editorial for this study. I thought it was terrific. The first author was Black, I think it was Dennis Black at UCLA. but they showed that this was this is something that could be disseminated and scaled up in communities. And I'm in the School of Public Health, so thinking about ways that we can affect populations, you know, people go to senior centers. Maybe that is a way that we can improve sleep on a broader scale using a technique that is not necessarily, it doesn't require a PhD and a license to impart. But they're definitely, and part of cognitive behavioral therapy for insomnia can include arousal reduction strategies like diaphragmatic breathing and meditation of the sort you're talking about. So it's a little separate, but it's also sometimes put into a CBTI framework. Barry, I think you had a hand up at one point.
I did, just very quickly. Are there any studies that you might know of that talk about exercise as affecting that relation between these negative outcomes and sleep deprivation?
So there are studies, and I'm working with a postdoc currently named Daniel Callow at Hopkins, and we are doing studies. And so people study, a lot of the time people have studied physical activity or exercise and cognitive health, right? And then they've studied sleep and cognitive health. But these things are related. As we spoke about earlier, we think that one can promote the other. So we are doing a series of studies showing, really examining that. And we are seeing that the association among, or of physical activity with some cognitive outcomes can differ by the sleep quality. And I forget all the details, and there have been we've published several papers recently. But I'm very interested in that interaction. I think that's really important. So I'm glad it's of interest. I can send you the papers. Thanks for asking. How long am I allowed to go before you pull? 5 more minutes, roughly? All right. Let's see. Does stress play a role in sleep fragmentation? Well, you know what's interesting? Oh, why? Well, let me go. I already answered a question from the person who asked this one, so I'm going to ask, why is more than 8 hours sleep per night potentially bad? It's probably not. If you see an association between long sleep and a health outcome, whether it be dementia or mortality or something like that, it's probably not that the long sleep itself per se is causing the outcome. It's probably that the people who are taking, are sleeping for a long, long time have something else going on, whether it be a bunch of medical problems that are keeping them in bed and more vulnerable to, falling asleep, or, your body, when we're sick, we know that our bodies tell us to sleep. So that's so we have to be careful about interpreting it as a causal factor. But I think that the recommendation of not having the recommended sleep duration be as long in older people as in younger people, 7 to 8 hours as opposed to 7 to 9, is because we don't know that it's not causal. We just don't know why it would be if it were. So I think it's more of a cautious move on the part of sleep societies. Yeah. So that was that. And does stress play a role in sleep fragmentation? So sleep and stress can work in bidirectional manners. If you don't get enough sleep, we tend to be more irritable, emotionally responsive or reactive. We tend to snap at people we love, potentially, right? And then on the other hand, if we're experiencing a lot of stress in our lives, we might be up worrying about our children, as was volunteered. So it's interesting. These things can, and then, so you basically experience more stress as a result of sleep deprivation, and stress can lead to poorer sleep. So it can be a cyclical issue. Yes, sir. Yeah, it's you.
How do you explain the differences between the amount of sleep that different people need, particularly those people who seem to get by with just 2 or 3 hours a night? And there are some, I believe, who don't need sleep. So the question is, how do you explain rest? they don't need rest? What is it that happens when you do sleep that most people need, I guess, is another way of turning it.
Yeah, so there are people who are short sleepers. 2 to 3 hours would be far, far outside the norm of what people, if for someone to be comfortable with that, you do hear about it. And there are genetic factors that play a role here. There are, so genes are important. And you will see in running in families, you'll find people who don't sleep well, just in families, or they might be very short sleepers and so on. But so we don't know exactly what sleep does, but there's a great quote that I think came from, was it Alan Rechtschaffen, who was one of the people who developed the scoring system for sleep. And it was either him or someone else. But the important thing is that it went along the lines of, if sleep, given it makes us vulnerable to getting eaten by predators, getting robbed or murdered or whatever, right? And we do it for 1/3 of our lives, if we're lucky, right? If sleep does not serve an absolutely vital function, it's the biggest mistake evolution ever made, is the statement. I think it's Alan Rechtschaffen, I could be mistaken. But there's another, we know that if you sleep deprive an organism, it will, like too much, it will die. And we see that with animals, animals, when they are deprived of sleep, will forego food and water to sleep. It is really essential. And we live in a society that has historically denigrated sleep as a waste of time, and as something that lazy people do. And, if you really want to go out and get them, the early bird and all that, right? And I think that we have to start paying attention to the fact that it is absolutely vital. And we don't know, but the thing that's so interesting is we don't know exactly why. We just know things sort of fall apart if you don't get enough. There are some people who seem to do remarkably well. I don't know whether rigorous studies have been done of those very short sleepers to see what happens to them. They might do just fine through through middle age, and then they keel over. Who knows? Who knows? But I'm not going to try. I enjoy sleep too much. That's great. Thank you so much for having me.